Welcome, and today's lesson will be about photosynthesis and more specifically, the light-dependent and light-independent reactions that make up this complex process. But before we talk about photosynthesis, it's important to talk about the chloroplast, where all of this takes place. And the chloroplasts are organelles found in plant cells that are responsible for photosynthesis, the process by which light energy is converted into chemical energy in the form of glucose and many other organic molecules. And chloroplast contains pigments called chlorophylls that are able to absorb light energy from the sun and use it to power the photosynthetic reaction. And it does this for visible light, with it using red and blue light the best, and green light the worst, since it being green, it ends up reflecting the green light and is unable to use it. The ability of chloroplasts to absorb and utilize light is essential for plant growth and survival. It allows them to generate the energy and nutrients they need to carry out metabolic processes. The amount and quality of light that plants receive can have a significant impact on their growth and development, as well as on the quality and quantity of the crops they produce. The more light a plant receives, the more photosynthesis it can do, the more glucose organic molecules it has that allow it to grow. The first part of photosynthesis is the light-dependent reaction, and this starts with the chloroplast absorbing light, and the pigments, specifically in the phycoid membrane of the chloroplast. The pigments, such as chlorophyll, absorb different wavelengths of light and transfer that energy to the reaction centers. This absorbed light energy is used to excite electrons in the reaction center of photosystem 2, which is named since it was the second photosystem to be discovered. The excited electrons are passed on to an electron transport chain, which generates a proton gradient across the phycoid membrane. This creates a different amount of protons, which then attracts them onto different sides of the phycoid's membrane. In order to replace the electrons lost by photosystem 2, water molecules are split into oxygen, protons, and electrons by a process called photolysis. And those protons are really just the hydrogen atoms that were in the water, but there's no electrons, so they end up being positive instead of neutrally charged. The oxygen is then released into the atmosphere, while the electrons and protons are used to replenish the electron supply to photosystem 2. The proton gradient generated by the electron transfer chain is used to power the ATP synthase, an enzyme that synthesizes ATP from ADP, an inorganic phosphate. The electrons from the electron transfer chain are then transferred to photosystem 1, where they are excited by a second input of light energy. These high-energy electrons are then used to reduce NADP plus to NADPH. This reaction is important as it produces both ADP and NADPH. These are both important molecules as they power the light-independent reaction, which is then going to produce the glucose and other organic molecules that the plant is going to need to function. And here's a more thorough diagram. As you can see, in the phycoid membrane, on top we have the stroma, which is the space inside the chloroplast, and underneath is the phycoid lumen, which is the space inside the phycoid. And light hits photosystem 2, causing the electron to move through the chain, and water being broken down into O2 and a proton. These move through, creating a gradient, attracting more protons, which then allow for the reduction of ATP, and photosystem 1, having its own electron transfer chain, helps to generate NADPH, which will then move into the light-independent reaction, which is also known as the Calvin cycle, which the first step of the Calvin cycle is the fixation of carbon dioxide into an organic molecule. The process is catalyzed by the enzyme Rubisco and produces a molecule called 3-phosphoglycerate, or just 3-PG8. And Rubisco is one of the most important enzymes, as it is the main way of generating glucose and other organic molecules, and it's one of the most abundant enzymes on Earth. The 3-PGA molecules are then reduced by a series of enzymatic reactions that use the NADP and NADPH that was generated in the light-dependent reaction. And the end product of this reduction process is the molecule G3P. Some of the G3P molecules are then used to synthesize glucose and other organic compounds needed by the plant, while others are recycled back into the starting material of the Calvin cycle to continue the cycle. Because it is referred to as a cycle, it does have to be able to continue on its own, and this is how it does it, by recycling this G3P. Overall, the light-independent reaction of photosynthesis uses the energy stored in ATP and NADPH generated in a light-dependent reaction to fix carbon dioxide and synthesize organic molecules, which are essential for plant growth and metabolism. And here's a more thorough diagram, as we can see carbon dioxide entering into the Calvin cycle, where 3-PGA is produced, and then through the use of the ADP and the NADPH, we get G3P, which will then allow us to make glucose, and then it travels through, being recycled again with the help of ADP into your RUBP, which will then continue the cycle. Photosynthesis is a complex process by which plants and other photosynthetic organisms convert light energy from the sun into chemical energy in the form of organic molecules. The process of photosynthesis can be divided into two main stages: the light-dependent reaction and the light-independent reaction, also known as the Calvin cycle. The light-dependent reaction takes place in the thylakoid membranes of the chloroplast and involves the absorption of light energy by pigments such as chlorophyll, the generation of a proton gradient across the membrane, and a synthesis of ATP and NADPH. The energy-rich molecules are then used in the light-independent reaction, which takes place in the stroma of the chloroplast. During the light-independent reaction, carbon dioxide is fixed into organic molecules using the energy stored in NADP and NADPH. And these molecules are then used to synthesize glucose and other organic compounds that are essential for plant growth and metabolism. The Calvin cycle involves a series of enzymatic reactions that use the energy stored in ATP and NADPH to synthesize organic molecules such as G3P. Overall, the two reactions of photosynthesis work together to convert light energy into chemical energy in the form of organic molecules, which are essential for plant growth and survival and the sustenance of life on Earth. Thank you for watching.